From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 40, recorded on May 2nd, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hey, Vincent. Hey, everyone.
How's it going?
Good. We're having flooding issues because all the snow has to melt. So, been dealing with that. My house is fine, but yeah, looking forward to some break in the weather.
Also joining us from New York, Tim Chung.
Hello. Hi, everyone. Good to be back. No flooding issue here.
And from New Orleans, Vivian Morrison.
Hey, everyone. Good to see you. I think I missed the last round.
You did.
Yeah.
You're busy. You had 10,000 grants to write or something.
Yeah, which is actually ironic since I wasn't the PI on any of them. So that's going to change. That's one of the things that has to change.
Let us know how that works out.
All right.
Maybe Ori will pop in any moment, but we're going to start. And today is Tim's turn to educate us. What are you going to tell us about today, Tim?
I think educate is a strong word. I'll just say talk about some stuff. So today we're actually going to talk about two papers, both on dopamine, since I work on Parkinson's disease. Seems like I'm kind of contractually obliged to look at these papers. And they are fascinating papers. That's why I'm going to talk about them. So the first paper we're going to talk about, by the way, you guys will ask me questions if there's anything unclear. Just butt in it whenever. The first paper we're going to talk about is on dopamine system on the dorsal striatum. So the dorsal striatum is, by dorsal, it's kind of like towards your back, towards the top of your head. That's dorso. So think dorsal fin of a dolphin. And that part of the striatum is the first, it receives dopamine from the midbrain, and that dopamine signal is the first to go in Parkinson's disease.
What is a striatum?
So striatum is part of this thing called the basal ganglia. So it basically, I'm just explaining, it's like dictionary.
He's just using more words.
But this,
so
the basal
it's deep in the brain.
Deep in the right. It's a bunch of. Sorry, Vivian.
Oh, I was just going to say that, like, it's deep in the brain and it does a bunch of functional stuff that Tim talks about, but it has a lot of relays for neurons coming from different places, and the reason it's called the striatum is that when you cut into it, it has these like striations.
Yes, super duper striated.
I just was recently dissecting a brain out of an early postnatal brain from a mouse pup with a student, and pulled the cortex away from the midbrain, and you can see the different, like the structure of the striatum in there, in the flesh, and see all the little striations.
Huh. Yeah, it's
it's in the middle there.
Yeah,
it's
it kind of looks
not the top curly part.
Right, not the cortex. It looks kind of like marbled when you slice it, and that's because your cortex, which covers the basal ganglia, your cortex actually sends axons down to your spinal cord, and those axons actually travel through the striatum, and those axons are myelinated. So when you slice a striatum, you see a lot of like axon bundles going through it, so it kind of looks striated. So that's why people call it the striatum. It's part of the basal ganglia. But it's well known that it is important for movement. So dopamine neurons from the midbrain projects to the striatum, and when you lose those dopamine, that is when you get Parkinson's disease, and you lose the ability to move. So it's very important for clinical reasons, but it's also important for other interesting reasons. For example, as we were talking before the recording started, a lot of drugs actually, a lot of medication and drugs target the dopamine system. So, for example, amphetamine and also cocaine target the dopamine system, and some of them can be addicting. And for that system, people think much of the effect is via dopamine's effect on the ventral striatum. So this is striatum that is more on the bottom side of the brain. And they receive a dopamine neuron from a slightly different projection called the ventral tegmental area. So we'll actually talk about both of those today. First, we're going to talk about the dorsal striatum and movement. And this is a paper from Bob Datta's lab. So the first author is Jeffrey Markowitz, and the last author is Sandeep Robert Datta. And if I remember correctly, Bob Datta actually was on TWiN a while ago during COVID, where you guys talked about him sequencing the nose and all the kind of neighboring glial and accessory cells and how that might explain the loss of smell. But for this paper, we're going to completely change gear. So his lab does many different things. This paper, we're going to talk about this thing that they did called motion sequencing. So motion sequencing is a technique that they developed where you can basically take a video of the mouse and kind of pass out what the mouse is doing into different, what they call behavioral syllables, which are different modules. So I better explain this a little bit more. Often, kind of historically, when neuroscientists and also psychologists study behavior, what we like to do is we give a mouse like a lever, and the mouse can press, or pigeon, or an animal, like a button to press or a lever to press. And when they press a lever, they earn some food. And we can look at how they interact with the lever. And this is a way of kind of for us to describe, to look at what behavior is changing. So a way of looking at learning and how the brain works. So a good thing about that is that you designed what the mouse is doing, like, or the pigeon or the animal. You kind of have a very specific task for the animal to do. A light comes on, the mouse has to press a lever, get some food. And you analyze the lever pressing as a behavior unit. The downside of that is that you lose all the other things that the mouse might be doing in the environment. The mouse might be exploring, it might be sniffing in the corner, it might be checking out where the food is. So an alternative approach is, which these authors did for this paper, is just put the mouse inside a bucket, an arena, without any reward, and just look at the mouse and see what it's doing, and ask what also is going on in the brain, and trying to associate what's going on in the brain with what the mouse is doing.
This is sort of like National Geographic for mice.
Exactly.
More a naturalistic kind of behavior.
Well, to the extent that you'd naturally find a mouse in a bucket.
Yeah, right.
Yeah, I was going to say there's very limited input there.
But this is not, Bob Datta's group's got some really sophisticated analysis, but this is, I think this is a big movement now in neuroscience. And there's a lot of different versions of this sort of analysis where you have machine learning. You basically train a network or a computer to sort of break down these modules and use that as a way to sort of discretely say what's happening in behavior, as opposed to, as you said, which are sort of more basic ways of assessing behavior, was to say, well, is the mouse pressing a lever?
Right. And you don't look at anything else. You just pay attention to the lever. So that's exactly, as Jason said, that's exactly what these authors did, was they took videos of mice. They actually used a 3D camera based on, they used like a Kinect, like a Microsoft Kinect, which was like an old gaming console camera that people used to be able to do, where you use gesture to control like video games. You can do that to a mouse, and you can capture exactly what a mouse is doing. And you can essentially use machine learning to break down different modules that are kind of almost not quite kind of similar to stereotypes. So picture if you're not a mouse, you're going back to being a human. Each time you reach for a cup and take a drink, it's kind of, you're doing the same action. Even though you're taking different sips of a drink, it's kind of similar. And, there are other instances like, when you look at your watch, it's a very stereotyped like movement. So it makes sense to assume that maybe a mouse's behavior can be broken down into these different behavioral modules, or as they call it, syllables. And kind of more complicated action is actually kind of, you can think of stitching together these different behavioral modules so that the mouse is, exploring the environment. So that's, I won't go into the details, but they use machine learning to basically chop up mouse's behavior into these little syllables. And they can, in the previous publication, they can actually show that if you pick out the same syllables and line them up, the mouse, you can show that it is kind of somewhat stereotyped. And what's interesting is that it is kind of like, imagine you're playing tennis, for example. In tennis, it's very obvious that you can have different behavior modules. Like, you can have a forehand swing, and a lot of forehand swing would look similar versus your serve, which would be like, smacking the ball over your head, versus like a backhand. So like, it's almost actually like a video game where you have different actions that you can select, and you press a button, and like, if you're Super Mario, you can press a jump button, and all the jumps kind of look similar. So maybe the idea is that maybe the mouse's behavior is kind of like that, where there are different behavioral modules it can select, and then it stitches it together. So the question they're asking is, since the mouse's, well, they did show that the mouse's behavior can actually be broken down to these different modules. The question they're wondering is, how does dopamine, which has been known to be important for movement, for example, in Parkinson's disease, how does dopamine change? Does dopamine explain any of these behavior? And this is an interesting question because for historically, for a long time, what scientists have been interested in and have assumed is that dopamine is kind of correlated with just movement, how fast you move. And that's largely driven by the fact that if you have too little dopamine, for example, in the case when you have Parkinson's disease, your movement either tend to slow down or just completely stop. You can't initiate movement. Whereas if you have too much dopamine, for example, a mouse that is on a lot of amphetamine or on cocaine, they would run around the chamber, kind of be all hyper. In human, if they're on cocaine, they tend to talk a lot, apparently. So you can just have a lot of behavioral output, let's say. So people have assumed that maybe if you were to look at how much dopamine is being released to the striatum, you would just get, the faster you move, the more dopamine you have. And this is actually one of the questions that these authors addressed. So we're diving into the paper now. So one of the first questions that they addressed is to look at dopamine release in the dorsolateral striatum, actually. So dorsolateral striatum receives a lot of input from the cortex, from the motor region of the cortex, as well as from dopamine projection from the midbrain, from this place called the substantia nigra, which is the bit of midbrain that is lost in Parkinson's disease. So they recorded the dopamine signal in this dorsolateral striatum using an optical sensor that fluoresces whenever there's dopamine there. And they first asked the question, before breaking the mouse down into these behavioral modules, we're just going to look at the mouse moving around the chamber. Do we see the phenomenon where the faster the mouse moves, the more dopamine is released? And they did the correlation, and they found no good evidence that that is true. If anything, there might be a slight negative correlation, so that the faster, at least in small time scale, if you look at very fine time scale, like on the order of seconds, if a mouse is moving fast, there tends to be maybe less dopamine. When the mouse is stopping, there might be more dopamine, which is counterintuitive. So the authors then decided to break it down to the behavioral modules that they managed to use machine learning to extract, and ask the question: Can we get if we align these behavioral modules to the dopamine signals, do we get something that makes a bit more sense, so that we can explain what dopamine is doing? So I should explain, like, dopamine is fluctuating very quickly in the dorsolateral striatum as the mouse is moving. It's kind of I think some of these peaks are on the order of like 50 to 100 milliseconds, so it's quite fast. So what the authors next did was to break up the behavior to all these different behavioral modules, and then lined up the dopamine signal to when the mouse transitions from one behavioral module to another one. So actually, I should talk about the example of the behavioral module that the mouse is doing. What is the mouse doing when you put it in a bucket without any reward? So what they break it down to is the mouse can do things like running. It can also turn left, turn right. It can rear up, so stand up like a meerkat. It can kind of dip his head and sniff around. So kind of what you imagine a dog would do in a park, but shrink it down to a mouse, and then it's in a bucket.
Hey, Tim.
Hello.
Can I ask just one question?
Please.
The dopamine reporter, and maybe you said this, but I spaced out. So it is like, is it like what is it exactly? Like, are we looking at like we're looking at binding of dopamine to a receptor, or is
exactly?
It, okay. We're not looking at like activity of a dopaminergic neuron. We're actually we're indirectly looking at the amount.
Exactly. We're indirectly looking at dopamine release. It's a fake dopamine receptor that, instead of acting like a dopamine receptor and signals downstream, it instead, when it binds dopamine, there's a conformation change and the molecule becomes more fluorescent. Yeah.
Cool.
And it's genetically encoded. So it's sort of a breakthrough in the last 5 years where you've had these genetically encoded reporters of neurotransmitters. Before that, the best way, or the most sort of accurate way to assess them, was to use electrodes that you'd have to stick in the brain. And then, sometimes you'd have the good resolution, but you wouldn't be able to know which cells are actually releasing the neurotransmitter. So that's, I think, a big breakthrough. And so, if people have heard of dopamine, they've probably heard of it being involved in the reward system as well as movement. And there's always been this idea that, you get rewarded and you get this massive burst of dopamine. But it's not that simple.
That's the second paper that we'll be talking about. But exactly, like Jason said, this is a new breakthrough to look at dopamine using light. Apparently, you have much better signal to noise, even compared to using kind of electrode to detect dopamine. And yeah, so that allows you to really see these fast fluctuation and align it nicely to when the mouse is transitioning from one behavioral module to the next. I should note that these behavioral module, it's amazing that they can do it, because the mouse doesn't tell you, hey, I'm doing a new behavioral module. You really have to use computation to guess that the mouse is doing it. But even though the researchers are using computation to kind of segment out these different behavioral modules, when they align the dopamine signal to when the mouse goes from one module to the next, they see a very clear signal where at the end of a module, before they transition, there's a slight dip of dopamine. And right after transition to a new module, there's a peak of dopamine. So it doesn't matter what the mouse is, it turns out it doesn't matter if the mouse is going fast, going slow, or different variety of behavior. If you align it to the behavioral module transition, you see this very nice signal of dopamine dipping and then rising, which is very interesting. So what they And they can also do it the other way around, where you can ask the question, if I were to align all the behavioral transition, is there any changes in dopamine before and after? And they also see this dopamine signal if they align it that way.
They need to figure out how to do this in humans, because I was just thinking about, if you have like kind of chains of like behavior, either habits or not. Like, so for example, in people who have ADD or ADHD, they'll start one behavior and then, shift rapidly to something else. And I wonder if if you would see, and they have an issue with dopamine, not in I don't know if in the striatum, how much it is. I think we talked about that briefly before, but.
Is this ADHD patients before or after medication?
Before. So that's why I'm like, oh, that's interesting. Obviously there's more granularity here. And when we're thinking about mice or humans, or especially with humans, there's more involved, like, what are the, which receptors are expressed where, because like we know that there are certain kinds of dopamine receptors that are kind of more concentrated in certain brain regions. So, there is some additional like gray area,
right,
intended gray matter that we don't know, but it would be, can they do something? Can they do anything like this in like functional imaging? With humans?
People have looked at dopamine release in human. The way they do it is they have a kind of a they have some sort of
ligand,
pet ligand that would bind to a postsynaptic dopamine receptor. And if there's dopamine release, those ligands get displaced and you see a change of signal. So people do that. But since you're going to be in a PET scanner, you won't be doing weird behavioral module. Like you wouldn't be
like, oh, there's the laundry. Oh, I should probably cook this, or that kind of stuff.
Your question of whether I'm going to interpret your question as whether dopamine's involved in making behavioral module more kind of stochastic is a question we're going to come back to in a couple of figures.
Okay.
The other thing that's weird about dopamine or in quite a few of these neurotransmitters is that we still don't actually know at the molecular level how dopamine gets released. It's not being released through canonical synaptic vesicles at synapses, for example, which is what we think happens for almost every other big neurotransmitter. And so the localization and spatial domain at sort of the subcell level was still a mystery.
Oh, do you mean that they are released at like these bulging in axons?
Right.
Like these
they're not really like real synapses, and so
right.
that's
this
is amazing.
And they like release by volume transmission, like they just get spilled out. But they get spilled up, they get taken back very quickly by a dopamine transporter. And when they don't get taken up quickly, for example, if you are on cocaine or Ritalin, then it really changes your behavior.
Wow.
Well, guys, I just am completely changing my research focus now, and I'm going to study that.
Amazing.
Yes. But going back to the paper, so they found at these transition bits from one module to the next in the mouse. So like when the mouse go from rearing going back to the ground, for example, and then to sniffing, at these boundaries between behavioral modules, there are these dopamine signals. So the next obvious question to ask is, do these dopamine signals come in different shapes that are predictive of what the behavioral module is, right? So for example, maybe the dopamine signal that comes with the mouse rearing looks very different from the behavioral module, looks very different from the dopamine signal that comes with the mouse walking forward. In some sense, maybe dopamine is still coding the kind of like the actual movement of the mouse. It can be 3D, 2D, sniffing, that kind of stuff. So the authors classified these behavioral modules into different groups. For example, rears, the mouse is standing up, or the mouse is grooming so licking itself and cleaning itself, versus the mouse is walking or scrunching up like doing the worm. And And they found no differences actually in the dopamine signal. Like they couldn't tell the difference between the dopamine signals associated with these different behavioral modules.
At what level are we saying differences? Like magnitude, patterns?
Exactly. So they tested a bunch of different things. First, they just looked at the magnitude and they couldn't tell the difference. Like it's not like when you rear very tall, you have a bigger dopamine spike than when you are dipping your head low. So nothing like that. But they also looked at the entire dynamics of the dopamine, so the entire waveform of the dopamine, so to speak, the signal, how the signal evolves in time. And they still, so for that, they use a machine learning mechanism to see, can we train a computer to recognize, if I give you this dopamine waveform, can you predict it corresponds to this particular behavioral module? And they couldn't do it, which is, in a sense, a negative evidence for a tight association, but it's still evidence that that there's no tight association between different behavioral modules and the way dopamine is signaling.
Wouldn't it have just been so easy if the answer was yes?
Well, that's
what
what.
I was kind of like, oh, and then I was like, so what's coding for what the behavior is?
Oh, but what's interesting is if you were to, so the same first and last authors, so Jeffrey Markowitz and also Bob Datta, so the same lab, had a previous paper where they looked at the neurons that these dopamine projection talks to in the striatum. In these striatal neurons, they actually receive input from the cortex as well. So the cortex kind of talks to these neurons in the striatum. Dopamine also comes in at these behavioral transition. So these neurons that receives all these input, they do seem to code for what the mouse is doing.
Right. Okay. That makes a lot of sense.
Right. So that kind of suggests. So that raises the question. In that case, what does dopamine do? What is quite interesting?
They're dopamine sensitive neurons, but they're not.
They are dopamine sensitive neurons,
but they're not releasing. They're not dopamine. They don't release dopamine themselves, right?
No, they sense dopamine, and dopamine does things on them. So they are the receiver of dopamine.
So like when so the thing that I'm kind of like looking for, that I think that I was thinking was going to happen, was that, oh yes, there was going to be a particular waveform or series of, action potentials or something like that was going to code, is going to be in part with like which neurons were activated, was going to code for the behavior for the syllable. But like obviously the dopamine's not doing that. But if you looked at the output of the neurons, they said those neurons
in the striatum, like the postsynaptic neurons in the striatum.
Yes, if you looked at like what they're doing, would you then be like, oh, could you then do the thing that I was hoping we could do?
It. Yeah, they can then
you could like
decode
look at those things and be like, this is indicating that this behavior is happening, even if you weren't to see the behavior yourself.
Yep, exactly. They could do that.
Okay, cool.
In a previous paper, they showed that they can do that, which is quite interesting. So it just suggests that dopamine is doing something other than exactly coding your precise movement. It is doing something else.
Yeah, I mean, it's kind of like your visual system in a way. Like those first order neurons are not coding what you're seeing. They're just coding if it's contrast or if
edges,
it's like shape, in this direction or in this direction.
Right.
And it has, and then that information gets relayed into overlaid on top of some other structure that involves different neurons, different neurotransmitters, and then eventually we lead to
exactly. So so early order kind of visual neurons can code for, like, even in the retina, can code for things like edges and stuff and boundaries. And here it seems like dopamine is especially firing at the boundary between behavior transitions.
Boundaries are so important in, interpersonal relationships and brain function.
That's right. We need a behavioral segmentation. We need a interpersonal boundary segmentation algorithm.
This is how dopamine neurons feel about it.
What's really funny is that they showed that they were comparing like different dopamine waveform, and they found two different dopamine waveform that look very similar, but they code for completely different behavioral modules. So like they both kind of go down and they go up. And one of them is the mouse turning left. So the mouse does something, turn left. And the other one, these researchers label it investigate, which is, I don't know what that means in a mouse. I kind of picture the mouse kind of in a Sherlock Holmes hat, like with a pipe and a magnifying
just
glass
exploring.
on the floor.
Did you look?
It's like,
but investigate,
is
that more directed? Like I'm going to spend a certain, is this like novel object kind of thing
mouse,
where you spend more time doing
the
something
mouse slows down, and
thing?
I'm guessing it's sniffing the bedding or the ground a little bit. Maybe it's sniffing the poop.
Yeah, I mean,
but they just
that's
call
one
it
critique of this method is that, the computer doesn't care what the behavior is. It's just coming up with these modules. But then the humans are trying to interpret what the behavior means. And that's where you sort of run into issues where it's, one, is this a real behavior? Two, what can we actually know what the mouse is doing just purely on this little like motif?
Yeah, that's
and that is why humans will always be necessary in science, right?
Yeah, better than
maybe the
we're
mice
never going to be
problems.
replaced.
Are you sure?
I'm not, but I'm just, that was my cry.
Don't we have
hope.
AI at this point? Like if you just put in the prompt for Chat GPT-4.
Yeah. So we do on TWiP, we do a clinical case every month, and we have people we give them symptoms and signs, and people guess. They just now give it to Chat GPT, and it gets it right.
Oh boy!
Wow!
I also heard just last night that there's, and I know this was all secondhand, it's probably third hand, that now AI can like just based on like your brain. I don't know what it was like, what that means. It's like fMRI or I don't know what telemetry. That it can like tell what you were thinking, like people had to were telling themselves a story, and the machine was like, okay, and then it says like this is what the person is thinking about, and even though like the specific, and this might be related to dopamine actually, everything is right, but
they're probably measuring EEG
maybe,
or something like
that. It wasn't that the specific content. The machine couldn't come up with that, but it could come up with uncertainty. Like there was the story was like my wife went to the grocery store, and then she must have forgotten something. So she turned around and came back home, and she showed up earlier than I thought. And the computer came up with something like, my wife went shopping. Does she miss me? Which is like, she came back. So like, why did she come back? But it was like in both situations, there was some sort of uncertainty. And we know dopamine is involved in learning. So if you do something, expect one outcome and it doesn't occur and you're surprised, like that's very meaningful to your, and like, I think very clear in your brain. So
that's second half of today's paper.
But
the dopamine uncertainty was actually a previous TWiN where I talked about a paper from Adam Kapesh, and it was talking about whether dopamine causes you to hallucinate when you're overconfident, even though there's nothing going on. Go back. So I can't remember which TWiN.
Happens to me all the time.
Gosh.
But that was a different part of dopamine. That was like at the very tail of the striatum. So it's a different part of the striatum. Like the striatum is getting chopped up, getting segmented into these different regions. It's quite interesting. But going back to the paper, I got to wrap up this part of the talk soon to go to the second paper. So to quickly review, the authors found that the dopamine doesn't seem to code for the actual behavioral syllable. It's the mouse rearing, it's the mouse running forward. And in fact, if you focus on just one syllable, so for example, the mouse suddenly pausing, and then you look at the dopamine signature, you see, on average, the dopamine spike right after the mouse pause. But if you look more closely, you see actually there's like a wide variability of how high the dopamine spikes. In fact, sometimes the dopamine can go negative. So most of the time it goes positive, but every now and then it goes negative and spans a very wide variable range. So these authors are like, well, what's going on? Why is dopamine having such a huge, variety of signal, even to the same behavior module. So what they did was they correlated, they asked a question. Since we talked about this already, dopamine is known to be involved in learning, and therefore things like drug addiction. Maybe for behavioral modules that has very big dopamine spikes right afterwards, those behavioral modules would tend to occur more frequently afterwards. So what they did was they picked out a single behavioral module, let's say the mouse rearing, and they asked the question, they divided those, essentially did a correlation for those trials, instances of the mouse rearing that comes with a very big dopamine spike. Am I more likely to see more rearing afterwards versus those instances when the mouse is doing the same rearing, but it's a very negative dopamine dip? And they found, yes, indeed, when the mouse is rearing and the dopamine spike afterwards is very big, then the mouse tends to rear more. Whereas if the mouse rears and the dopamine dips, then the mouse tends to rear less in the future. And they can even ask the question, well, how far in the future do I see this correlation, this association? And they found that it has a time constant, so it decays. Like the influence of this dopamine spike into the future decays as a function of time. And they found it would increase the probability of you doing the same behavioral syllable again for about roughly 80 syllables, if that makes sense. So like if you reached out and, if you're playing tennis and there's a big dopamine and you do a nice foreswing, there's a nice dopamine spike, you're more likely to redo that for like the next 30 seconds. But
why would there be a bigger dopamine strike there? Just because you're like, oh, look how great that worked?
That is a very good question.
Because I have actually one question about this. This is what you've just described. Was it, I can't remember. They looked at this spontaneously, right? And then they also recapitulated it by like inducing like a big dopamine spike, right?
Yep, that's upcoming.
All right, okay, great. I just wanted to make sure that that's not the one you were talking about, because then it got me thinking like, well, why are any of these behaviors in this bucket having a big dopamine spike? Like,
right.
What is it about having reared that was?
Why is this rear better than the last? Yeah, why is this rear more better? Should be more reinforced. Like give rise to a better dopamine than the last one. And
I think I think in humans, it's probably a lot easier to come up and like, well, obviously that's why that we all can say, oh, it's more pleasurable to do something this way than that way. So of course this is gonna. I mean, that's all based on learning too. So it's kind of a chicken and the egg situation. But yeah, that's just something I would throw out there, which is like, when we're describing like taking a drink of water, or you're having a cocktail, for example, and it's 5:00 somewhere. So if you're drinking a cocktail, you're repeatedly doing it, there's like a reason, there's a why that like makes sense to all of us. But what is that for the mouse?
Well, there must be something. We just don't know what it is, right?
We don't know what it is. I think
hopefully there's something. If there isn't, then that would just be, I wouldn't know how to interpret that.
Probably no. I don't think the dopamine's randomly being high or low, right? There's some other thing. Maybe the mouse saw something in the corner that was unusual, and it's hard to know. We won't know that. Yeah.
This paper, they talk about it in the discussion, and that's one remaining outstanding question, is that we do not know what controls East dopamine.
What's important to mice.
Exactly, or even humans.
But the fact that they do more rearing when the dopamine is high, that makes sense, right?
In the future.
Yeah.
They do more rearings in the future. It makes sense in that it kind of in agreement with what we know about dopamine in learning.
Okay.
But it's in some sense surprising that there are these very fast fluctuating dopamine even without any reward, which is the whole point of this experiment, is that the mouse is just in a bucket.
But maybe they've learned that by rearing or sniffing or any particular syllable, they've learned in the past that doing that behavior has a higher likelihood of leading to a reward, right? Exploratory behaviors might the mice might be exploring. Intrinsically be more like dopamine prone.
Yeah.
Right. But we do not know Yeah, it might be, but
Yeah,
we
we
really don't know
This
what.
goes back
reward
to
is
Vincent's
from
point that, the internal state of the animal is unclear to us. And
Yeah.
that's
It's-
what this is sort of saying, is that there are these internal states that we can't see really without going into the brain.
Yeah. And we're exactly speculating on internal state to use it to explain what dopamine is doing, which can be problematic. Like, for example, you can ask a question, why is Tim eating so much? And the answer can be, well, he's hungry. And then you ask, hunger is an internal state. And you say, well, how do you know Tim is hungry? Well, look at him, he's eating a lot. So it can be circular argument. So it's very important that if you're in an internal state, you have to look inside to see if there are things that explains this internal state. Anyway, going back to the paper, the next thing they looked at, which relates to what Vivian was asking earlier, is that they also saw when there's a big dopamine spike to a certain behavioral module, not only does the mouse would more likely to do the same behavioral module again, they would also be more kind of random in the next behavior they pick. So, for example, you can imagine like playing tennis. You might have very stereotyped choices of what you do. Like you might, serve the ball and then come to the net next every single time. But what they found is that if you have a big spike of dopamine when you're serving the ball, then your next thing you choose will be much more variable, much more stochastic in a sense. So this is kind of one way of kind of thinking about this is that mouse can be more prone to exploring if there's a big dopamine spike. But interestingly, they would come back to the original behavioral syllable more likely as well. So that's some of the things that these researchers found. And then the next thing, we're running out of time, so I'll very, very briefly talk about this, is that all I've talked about right now, just now, is all correlation. So these researchers next did the causality test where they actually stimulated dopamine at very particular behavioral syllables. So the way they have to do this is they have to have a camera, and then the camera will be hooked up to the computer, and the computer has to, in real time, kind of classify what the mouse is doing into these behavioral syllables. And then it would only pulse in dopamine, so that it would optogenetically stimulate dopamine neurons so that it would release dopamine. It would only do it at a specific syllable. And what they found, they recapitulated what they found in the correlational study. What they found is that when they did this, the mouse are more likely to do the same behavioral syllable again in the future. And it's also more chaotic in its behavior, kind of in the next choice of the behavioral syllable. So they basically managed to show that it is actually a causal relationship. And what's interesting is that they picked they had 6 different syllables that they picked. So they did the experiment several times. And these syllables ranges dramatically in how fast the mouse is moving. So again, we've been talking about historically, we think more dopamine equals more movement, but they can actually pulse in dopamine to train the mouse to pause. If they pick the pause syllable and pulse in dopamine during pause, then they can make the mouse pause more in the future. So that's something that really suggests to us that dopamine might be important not just for any movement, but for reinforcing and structuring these behavioral syllables and what the mouse is doing.
But you said that in the, so I found this interesting where they were talking about different time scales, like on the big, like throughout the whole trial, yes, administering a big bolus of dopamine on a particular syllable will, over the course of the trial, lead the mouse to do that syllable more frequently. But they also said that like in a very, very short time span immediately after, it was like whatever syllable was going to be paired with the one that they just enforced, it was like impossible to tell. It was like more variable than if you had it. Did I understand that correctly? I
might have explained it poorly, but usually before the mouse usually tends to have so the way the mouse strings together these behavioral syllables, you can think about it as you have a big Excel spreadsheet that write down all the different transition probability from one syllable to another one.
Mm-hmm.
And usually it can be somewhat stereotyped. They actually call it the grammar, kind of like the way we use sentence, and certain words would follow
And
other
how we
words.
learn languages.
Like how, exactly,
Our
like
brains,
it doesn't
yeah.
make sense to put like two verbs together, for example. So it might not make sense for the mouse to rear and then rear again because you're just going to strain your back, for example. So there are certain stereotype transitions as well between, in terms of how these behavior syllables are strung together. But if you pulse in dopamine at certain time, like at any syllable, it seems like, then the next syllable upcoming for the next few seconds would be less stereotype, like would be less
So it's
typical. The mouse is like more chaotic. It's almost like the mouse is like randomizing more.
Decrease the probability that of the syllabus, the syllable that in your spreadsheet is like most likely going to occur. The probability of that happening goes down.
Right. It's more even. Like the mouse is now exploring more syllables, like go to more different syllables in a sense.
That's interesting.
And that has been found.
Creativity?
Maybe. In terms of what you do, it's kind of like you put the mouse in an interpretive dance mode where it's just doing random things.
Oh, that's cool.
Yeah, I don't know. Like, there's actually an interesting question where these, the way we have been talking about it, these behavioral modules are almost like as if it's kind of innate. Like the mouse is just handed these bunch of behavioral modules, like the letters on a Scrabble board. You have to put them together to make words, whatever.
Right.
So, and the question is, well, can the mouse only learn new movement by stringing together these modules, or can you actually change these modules themselves? There's some hints within this paper that the mouse can actually learn to speed up and slow down these. Even within a module, they can speed up and slow down things. So suggesting that you can learn to change these modules. But it's unknown so far how mouse can learn to do new things based on this paradigm.
And I don't want to, I know you want to get to the other paper, but there was something in here saying that like and maybe you said this and I spaced out again, the amplitude of the dopamine spike would actually dictate like how fast or how slow, or it was something related to that.
It actually does. If you pulse in the physiological amount of dopamine, so like an amount of dopamine that the mouse naturally will release, then it doesn't actually change the speed that the mouse does. What it does is it reinforces the syllable. The syllable can be running forward, running very fast. The syllable could be pausing, and it doesn't really matter how fast the mouse is going. It would just reinforce that syllable.
Yeah, I thought there was something about like the speed at which something was done, because I wrote in here in my notes that when humans visualize something, we tend to move in our heads. Like, for example, if you're a gymnast and you're kind of thinking about the movements that you have to make to like run to the springboard, and then you have to do a series of flips in the air, that the length of time that you spend visualizing a certain part of it has to do with or how fast you move through a certain part of it depends on the complexity of the thing that you're trying to visualize. So even though you're going to be spending the majority of your time running down the ramp towards the springboard, like that's what most of your run.
Yeah.
The most complex thing is the jumps and the spins that are in the air. So you're going to spend more time thinking about those movements. And so I just wondered if, anyway, I'd written here in my notes something about how like the dopamine, that there were aspects of dopamine spikes or timing or whatever that would then influence how fast or slow something happened. And maybe I misread it, but then I was trying to think about, how does that translate to this kind of slow or fast visualization?
Yes. So
because just because something exists in the physical world, the same pathways can still occur when you're visualizing something or watching somebody else do something.
Oh, that I don't know if that's a good point. Like they can actually show mice watching other mice do things and earn reward and see if there's dopamine release. I don't even know if that experiment's been done. But but yeah, I don't know. We're going into a rabbit hole towards like replay inside the brain.
That's
but going back to what you said, like it's possible that the gymnast running all the way across the floor before it does a complicated flip, the whole running is one behavioral module. So you don't really have much, there's not much going on there. And then the different position adjustment that you do during the flip might be different behavioral module that you have to string together. But I don't know how a gymnast learns to do that and how that corresponds to the dopamine.
Lots of
falling. Would be a challenging experiment to do. But yeah, I think that, so basically we have, I'm going to wrap up the first paper here. Just quickly summarize. What we found was that we can break up a mouse's spontaneous behavior, even without any reward, just a mouse running around, exploring a bucket. The spontaneous behavior can be broken down into these somewhat stereotypical behavioral modules. And that at the transition between different modules, you see these dopamine spikes. And these dopamine spikes seems to kind of reinforce the behavioral module that it's associated with. So that more dopamine, if the behavioral module has more dopamine, then that behavioral module will be more used in the future. And the authors said that this might actually, we might have learned something about how the mouse structures its own behavior inside a bucket. So they speculate that the mouse is kind of reinforcing itself inside the head of what it should be doing and therefore kind of learning to adjust. What drives this, what teaches the mouse what dopamine to release is the open question that they don't know yet. All right, first paper. Second paper, I'm going to have to go.
Can you do it in 10 minutes?
I'm going to do it in 10 minutes.
Very interesting paper.
The first paper is meant to be the short one. Oh no. Second paper is
it's lots of math, right?
There's a lot of math. I'm not going to.
Okay, well then there's going to be way less talking from my end. So it's okay.
No, I'm not going to talk about
theoretical paper.
I'm not going to talk about any of the maths, but I'll just quickly go through the authors. So the first author is Huijiong Zhang. And the last author of this paper is Vijay Mohan Nambudiri. Sorry if I pronounce things incorrectly there, but they are from Ori's institution, so University of California, San Francisco. And the paper is called Mesolimbic Dopamine Release, Conveys Causal Association. And it's published in Science, I believe. Both these papers are published this year, 2023. So just now we talked about dorsal striatum and movement and a little bit about how it can act as a reinforcer to drive learning. The second paper, we're going to talk about ventral striatum, which is involved, people know to be involved in drug addiction and also learning. But the one thing that has been the predominant story for ventral striatum dopamine is that it encodes for this thing called reward. Well, people speculate that encodes for this thing called reward prediction error. So the discovery that led to this dominant theory now is that during the 90s, a researcher called Wolfram Schultz and his colleagues found that they were trying to train a monkey to, if I might have misremembered, but I think the story goes, they're trying to train a monkey to basically reach out into a box to grab a grape and to eat it. And they're at the same time recording from the dopamine neurons that project to the ventral striatum. So the dopamine neurons are in the midbrain in an area called ventral tegmental area. And originally, I think the expectation is that the dopamine neuron firing would correspond to the monkey reaching because of the Parkinson's movement thing. Instead, what they found was at the beginning of training, during early trials, dopamine would fire when the monkey is eating the grape. It would fire consistently when the monkey is consuming the grape. Not much consistent when the monkey is reaching for it. But after training as training progresses, the dopamine starts firing to like the sound of the box opening, the box that contains the grape opening, which is weird. And in a sense, this is kind of like Pavlov's dog, which is so I don't know, 150 years ago now, Pavlov would ring his bell. And every time he rings his bell, he would kind of hand over a piece of steak to his dog. So at the beginning, the dog would salivate to the steak because it's a steak, it wants to eat it. But you do it several times. Eventually, the dog would salivate just to the ringing of the bell because it's been associated with the steak. So the idea behind how to explain dopamine's response going from the reward. So at the beginning, before learning, dopamine would fire, at least the story goes, before learning, dopamine would fire when you give the animal or the human the reward. But after learning, it would fire to the cue that predicts the reward. The way we explain it is that dopamine encodes for the reward prediction error. So all the time, we're trying to predict what is going to show up in the environment around us. At the beginning, we couldn't predict that someone is going to hand us a steak or, an ice. As these researchers, they use the example of the chime of an ice cream truck and then the ice cream arriving. At the beginning, we couldn't predict that ice cream is going to be here. So dopamine fires to that. But as we learn that this ice cream keeps preceding, this ice cream chime or the bell keeps preceding the reward. Now it is the ice cream bell that is the thing that we cannot predict. So dopamine now fires to the ice cream bell. And in some sense, the reason that happens is because the positive value. So we assume the reason why we like ice cream and why the dog likes the steak is that those have intrinsic value. And the value event kind of propagates backwards through each learning. It propagates to the bell, to the cue that signals there's an upcoming reward. So that has always been the prediction of this reward prediction error hypothesis. And it is used in theories like reinforcement learning. And those are kind of the recent artificial intelligence tools that has led to like DeepMind beating the world champion in the game of Go and also chess. Like all these computer games has been completely cracked by computers now, because computers, you can also, this algorithm is basically a bunch of, it's an algorithm. So you can teach a computer to use these principle of predicting the upcoming reward to train it to kind of do things in the world and to, beat the game of Go. So in this paper, the researchers really put this reward prediction error hypothesis to the test. So the first experiment they did was, it is known that, so if you assumed the reward prediction error, then the very first time you see the reward, you should have maximum dopamine release. Because when first time you see the reward, you cannot predict, you haven't learned that the reward is going to show up. So dopamine's going to release is going to be maximal. Whereas if you keep presenting the reward kind of unpredictable, I misspoke. If you train the animal to, in this experiment, we're kind of in figure 4. We're jumping straight to figure 4. In this experiment, what the researchers first did was, one of the experiment the researchers did was they presented a tone, which is the conditioned stimulus, they call it, which is, the bell in Pavlov's dog. And then they presented the reward a few seconds afterwards. In the reward prediction error hypothesis, at the beginning, the dopamine would fire maximum to the reward. But as learning happens, the reward to the dopamine firing to the reward would decrease, and the firing to the tone would increase, which they did find. But what's interesting is that when they looked at when the mouse started licking, started responding, the responses to the tone actually appeared way, way later than when they first see the dopamine response, which, as it turns out, didn't really agree with the reward prediction hypothesis. I'm kind of explaining this a little bit poorly for this bit of the experiment.
I'm wondering if I'm wondering if we could just sort of, I mean, I think the biggest thing from this paper is not necessarily even the dopamine-ish stuff. It's just that the way we think learning happens has been this causal way in that, you know, something predicts the reward. So we learn that the tone predicts the reward. And what they're saying here is that, okay, actually what the brain is doing is that it's not that when it doesn't get that reward, it's telling us that there's an error. It's actually that the memory is retroactive. So you get the reward, and then the brain has to go back in time and think, okay, well, what happened before that reward to help me understand what the cause was? And that's that's the real sort of novelty here. It's usually we're just like, okay, cause and effect. But now they're saying actually the signal that the brain uses to learn the association is retroactive. It's a memory thing. You have to go back in time to say, well, what actually helped predict the reward? And that, to me, is really cool. I mean, that's basically rewriting potentially how we think brain works.
Yeah, exactly.
And then they were like, okay, well, and the evidence for this is that the dopamine signal is actually this retroactive signal rather than, you know, causative.
Yeah, I think the main, we're running out of time, but I'll just quickly talk about a couple of experiments that really is incongruent with a dopamine prediction error that people have been assuming. The first one is, you know, if in their case, once they have trained the mouse to expect this tone, to associate it with a reward, you see a dopamine firing to the tone, as you would predict. But then when they remove the reward, so this is called extinction, what you see is that if you keep presenting the tone again, the mouse eventually stops licking to the tone because the tone no longer predicts a reward. But when you look at the dopamine firing, the dopamine kept firing to the cue, which is completely not explainable with the reward prediction error hypothesis. And another experiment that they found that really couldn't be explained by this reward prediction is that the reward prediction error hypothesis predicts that when you ring a tone and then 3 seconds later you give the reward, what is going on inside the mouse's head is that the tone would elicit a certain internal state, and then there will be a bunch of internal states that kind of it transitions into until the reward. So kind of like what we talked about earlier in the first part of today's TWiN, the mouse is going through different behavioral module. Here, people usually assume inside the mouse's head, it is kind of when the tone arrives and is followed by reward after a delay. It steps through different stereotyped transitions so that it learns to expect the reward at the correct time. And if that's the case, what you would find is that as the mouse learns to associate the reward with the tone preceding it, at the beginning, the dopamine would fire to the reward, but at the next trial, the dopamine would fire to the state just preceding the reward. And then the next trial would fire to the state just preceding the reward. So in a sense, the dopamine signal kind of crawls backwards slowly towards the cue, because the cue is the eventual thing that predicts the reward that the mouse cannot predict. And what they found is that when they looked at the dopamine signal, there was this kind of crawling back of the dopamine from the reward to the cue, which completely cannot be explained by the reinforcement learning reward prediction error paradigm. So cut a long story short, the researchers came up with this model, as Jason said, where the mouse is in its head has all these kind of association between cue and reward. Basically, estimate whenever I see a cue, how much more reward can I predict based on this cue relative to background. They have an algorithm that does that, and that explains perfectly all these different findings. They have 11 tests in this paper. And yeah, it's actually, I think it's a very interesting way of showing that dopamine, the way we've been thinking about dopamine is been maybe needs to be revised. Yeah,
I think one of the cool things about this paper as well is that so we often in neuroscience, especially in biology, sort of poo-poo models, and it's just like, no, we need to do the experiment, we get the data, and then we make the model. This is one of the few high-profile papers where they were like, well, we have a model, it's based on previous data, but it's actually completely different to what everyone thinks the way it works, and here we're going to use that model to show it by then doing actual experiments. So it wasn't just purely theoretical. They proposed the model. The model in and itself was the opposite of what other models were out there, and then they took the data to reinterpret the model. So it's really cool, I thought.
Yeah, it's and the fact that they actually tested the original reward prediction error hypothesis. They worked out all the implication, and they did all these tests and show that in every single case it seems to fail.
Right.
So that's very, that's good, interesting stuff. Anyway, my apologies. I think I didn't do the second paper justice. There's a lot in there in the second paper that, for example, there's another cool, I'll squeeze this last one in. Imagine, so the very last experiment that they did, that's really cool, is they had tone 1, tone A, followed by tone B, followed by reward. And in the reward prediction error model, the kind of the canonical, well-accepted model, you would predict that if you block dopamine during tone B, the one in the middle, it would kind of jam up the ability for dopamine to go all the way back to tone A, the very first tone that predicts a reward. And they did that experiment using optogenetic inhibition of dopamine during the second tone. And they show that the mouse can both learn to associate the first tone with the reward. And the first tone actually does acquire this dopamine signal. And very briefly, actually, I should kind of conclude what the authors think dopamine in this case is actually doing. They think that dopamine's role in their model is it is a signal that this particular cue, or it could even be a reward, is something that is meaningful that I should learn about. Not only should I learn about this cue, I should learn about what causes this cue. Are there other cues in the environment, other things happening that consistently predicts this cue I'm looking at? And this dopamine signal can not propagate backwards, but can just be associated with other cues that predicts it. So the mouse has another giant Excel spreadsheet inside its head, and it's figuring out all these statistical correlations between all these cues happening in the environment. And dopamine is the one that
I think basically dopamine is telling the brain to pay attention.
Right. In a sense. Well, yeah, actually, yeah. So I would quickly...
There's like a thread through this particular episode.
Yeah, I would quickly shout out the work of Erin Calipari, previous TWiN host, who has a couple of beautiful papers recently published on the model that dopamine, instead of being a reward prediction error, is very important for the salience of a cue, should you pay attention to it, which also seems to kind of mesh well with this paper. So some kind of heterodox, kind of like a revolution, kind of new thinking about what dopamine might be doing.
Wow. I don't know why put so much into one chemical, dopamine, right?
Oh, actually, Vincent, that's a very good point. like why should the brain put so much?
Yeah,
like evolution should put,
yeah.
or why should we pay attention? I don't know why. So what's very interesting about, sorry, I just read the chat. What's really interesting about dopamine is it's very weird. If you have too little dopamine in the brain, you get Parkinson's, where you really want to move, but you cannot move. If you have too much dopamine in the brain, so this is patients on Parkinson's that are given dopamine medication. If they're on dopamine medication for a very long time, they can develop these movement pathology called dyskinesia, where the dopamine has become so dysregulated that they take these dopamine medication and they can no longer control the movement. They can't do anything but move. So too little dopamine, you want to move, but you can't. Too much dopamine, you want to not move, but you can't. So dopamine is a very critical substance that kind of seems to be sitting between your intention and the movement that comes.
And if you want an example of that, Michael J. Fox just had an interview where you can see exactly that effect, where he can't control his movements anymore because he's been on these medications for so long.
Yeah. And I don't know why we are piping our entire being from our intention to our output through dopamine.
Yeah. That was my question. Evolutionarily, why would that happen, right? It's odd. Usually it doesn't. All right. Thank you, Tim. That was really cool. That's TWiN.
Yeah.
Thanks.
Number 40, microbe.tv/TWiN for the show notes, comments, questions, TWiN@microbe.tv. If you enjoy our work, we'd love your financial support. Microbe.tv/contribute. Jason Shepard's at University of Utah. Jason Synaptic on Twitter. Thank you, Jason.
Thanks, guys. Thanks, Tim.
Tim
Chung is at New York University. Thank you, Tim.
Thanks, everyone. Sorry about the second paper. If listeners go read it, listeners who are interested in dopamine, highly suggest.
But you have to have access first.
That's a good point.
That's a problem.
Yeah, I'm at home, so I ran into that, but luckily I could sign in through my institution.
Yep.
But...
It might be on bioRxiv. Let me check.
Okay.
Vivian Morrison's at Tulane University. Thank you, Vivian.
Thank you for doing that awesome paper. It really kept my attention.
No.
I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.